When an axolotl loses a limb, an astonishing sequence of biological events is set into motion. Within hours, skin cells migrate over the wound, forming a wound epithelium that seals the injury. Beneath this covering, local cells begin to dedifferentiate, reverting to a more primitive, stem cell-like state. These dedifferentiated cells accumulate to form a blastema, a mass of proliferative progenitor cells capable of regenerating the diverse tissues required to rebuild the limb. Remarkably, the regenerated limb is indistinguishable from the original, and the axolotl regains complete use of it. Equally impressive is the axolotl's ability to regenerate large portions of its central nervous system, including the spinal cord and brain. In mammals, damage to the brain or spinal cord typically results in permanent loss of function due to the limited regenerative capacity of neurons and the formation of scar tissue that impedes recovery. In contrast, axolotls can fully regenerate neural tissue, restoring both structure and function. For example, if a portion of the axolotl's brain is surgically removed, neural stem cells near the injury site become activated. These cells begin proliferating and migrating to the damaged area, where they differentiate into neurons and glial cells. These new neurons integrate seamlessly into existing neural circuits, restoring cognitive and sensory functions without noticeable deficits. Not only is the architecture of the brain reestablished, but behavior and neurological performance also return to normal. This indicates that axolotls are capable not just of anatomical regeneration, but of functional restoration, a critical distinction when considering therapeutic applications. Axolotls belong to a group of amphibians called salamanders, which are renowned for their regenerative capabilities. However, no other vertebrate comes close to matching the extent and perfection of axolotl regeneration. Their remarkable powers have earned them the title of the world's most regenerative animal. The axolotl is a species of salamander native to Mexico. It is a neotenic organism, meaning that it retains its larval features throughout its life. Axolotls have a number of unique characteristics that make them an ideal subject for studying regeneration. First, they are highly plastic organisms, which means that their cells can differentiate and re-specialize into different tissues. This is in contrast to mammals, whose cells are more fixed in their specialized roles. Second, axolotls have a very efficient immune system that prevents infection and minimizes inflammation at the site of injury. This creates an optimal environment for regeneration. Finally, axolotls have a relatively simple body plan compared to other vertebrates, making it easier to study the mechanisms of regeneration. When an axolotl loses a limb, the first step in the regeneration process is the formation of a wound epithelium. This is a layer of skin cells that forms over the wound to prevent infection. Beneath the wound epithelium, a mass of cells known as a blastema begins to form. The blastema is a collection of undifferentiated cells that are capable of giving rise to any of the tissues hit in the amputated limb. The formation of the blastema is triggered by a complex interplay of chemical signals. One of the key signals is a protein called noggin, which is produced by the wound epithelium. Noggin is a member of the bone morphogenetic protein family, which are known to play a role in embryonic development. In the case of axolotls, noggin helps to establish the anterior-posterior axis of the regenerating limb. Other members of the BMP family are also involved in the regeneration process, such as BMP4, which regulates the growth and differentiation of cells in the blastema. As the blastema grows, cells begin to differentiate into the various tissues of the limb, such as muscle, bone, and cartilage. This process is guided by a set of molecular signals called morphogens. Morphogens are secreted by specialized cells at specific locations and concentrations, create a gradient that cells in the blastema can sense. This allows cells to know where they are located along the limb and differentiate into the appropriate tissue. For example, a molecule called sonic hedgehog is secreted from the zone of polarizing activity, which is a group of cells at the posterior end of the blastema. Sonic hedgehog creates a concentration gradient along the anterior-posterior axis, which helps to establish the correct number and arrangement of digits. Other morphogens, such as fibroblast growth factors and wingless-type MNTV integration site family members, regulate the growth and patterning of the limb bud. Perhaps one of the most remarkable aspects of axolotl regeneration is the way that they regrow their nervous system. In mammals, damage to the nervous system typically results in permanent loss of function. This is because neurons do not regenerate and the damaged tissue is replaced by scar tissue. However, axolotls are able to completely regenerate their spinal cord after injury. restoring both structure and function. When the spinal cord is severed, the two ends of the spinal cord first fuse together to form a structure called the neurocole. This structure provides a channel for axons to grow through. Neurons then extend their axons toward the growing edge of the wound. These axons are guided by chemical signals, such as netrins and semaphorins, which are secreted by cells in the blastema. As the axons approach the target tissue, they begin to differentiate. Sensory neurons differentiate into proprioceptors, which sense muscle movement and position. Motor neurons differentiate into alpha and gamma motor neurons, which control muscle contraction. Interneurons differentiate into various types of neurons that connect sensory and motor neurons. Once the axons have reached their target tissue, synapses are formed between the neurons. This allows the regenerated spinal cord to transmit signals and restore function. Axolotls are also capable of regenerating their heart after injury. In mammals, damage to the heart leads to the formation of scar tissue, which impairs the heart's ability to pump blood. However, axolotls are able to regenerate heart muscle cells, blood vessels, and the extracellular matrix of the heart after injury. This process begins with the activation of cardiac stem cells, which are located in the heart's crypts. These cells then proliferate and differentiate into cardiomyocytes, the cells that are responsible for pumping the heart. The regeneration process is regulated by a variety of molecular signals, including transforming growth factor beta, vascular endothelial growth factor, and fibroblast growth factor. In addition to their ability to regenerate their limbs and nervous system, axolotls are also capable of regenerating other tissues. They can regrow parts of their eye, jaw, gills, lungs, and kidneys. This remarkable regenerative ability makes them one of the most regenerative animals on the planet. There are several advantages to being so regenerative. For one, it allows axolotls to repair damage that occurs during their daily activities. For example, they are often preyed upon by birds and mammals, which can cause significant injuries. In addition, axolotls live in a very harsh environment. The lakes where they are found are often polluted and subject to drastic changes in temperature and oxygen levels. Being able to regenerate lost limbs or tissues is a great advantage in such an environment. So why can't humans regenerate like axolotls? The answer is not entirely clear, but it is likely due to a combination of factors. One possibility is that human cells are more specialized than axolotl cells, which makes it more difficult for them to differentiate and re-specialize. Another possibility is that the human immune system is too strong, and it attacks the regenerating tissue instead of protecting it. Finally, humans do not possess the same genetic toolkit as axolotls, which may limit our ability to regenerate. Despite this, there is hope that we can learn how to regenerate like axolotls one day. If we can understand the cellular and molecular mechanisms that allow axolotls to regenerate, it may be possible to apply them to humans. For example, we may be able to develop drugs that promote regeneration or gene therapies that restore the ability to regenerate. Axolotls are truly remarkable creatures. Their ability to regenerate their limbs, nervous system, and other tissues is unparalleled in the animal kingdom. If we can understand how axolotls are able to regenerate so efficiently, it may one day be possible to apply this knowledge to humans. Thanks for watching.